afternoon, everyone. I'm Guilherme. I'll be chairing the next session, so help me to give a warm welcome to Professor Julian Catchen. So Professor Catchen holds a bachelor's degree in computer science from Pennsylvania State University and a PhD in computational biology from the University of Oregon. Currently, he's associate professor of evolution, ecology, and behavior at the University of Illinois. He's the pioneer behind the STAX software, which is widely used in population genomic studies. And his aim is through the use of genomics and bioinformatic methods, Professor Catchen aims to study how genomes change over time, particularly and in response to major evolutionary events. Today, he will present a talk in the comparative and population genomics of an icefish that escaped Antarctica. Please join me and give a warm welcome again to Professor Catchen.
Okay, good afternoon, everyone. It's really a pleasure to be here. I really want to thank the student organizers of the conference who have been answering our emails for months and picking us up and dropping us off and just doing all sorts of logistics. I'm extremely impressed with their organization. So thanks to them and thanks for having us. So my name is Julian Catchen, and I have to say I feel a bit like a fish out of water. Sorry for the bad joke, but hopefully by the end of this talk, you all feel like fishes out of water, maybe. But I'm going to tell you a story about a fish from Antarctica. Actually, it's from the tip of South America that you can't really breed in a lab. And if you do make a cross, it'll take about 6 months to raise one generation, and you probably have to overwinter in Antarctica to do it. So this is kind of extreme genomics of a different sort, but hopefully you'll find this to be an entertaining story. And before I begin, I run a population genomics computational lab at the University of Illinois, and the work I'm going to tell you is the contributions were made by all of these individuals who are all members of my lab at different times. And primarily, I'm going to be telling you about Angel's work. And Angel's was a great student. He's now a postdoc at the University of Oregon. But everybody contributed, and I'll continue to highlight the different students throughout the talk. Okay, so one of the major questions of the day involves climate change, of course. And so there are a lot of funding in the United States going to trying to understand the effects of climate change, trying to understand, this is an example of how species numbers will change over time in the oceans under different climate scenarios. This is a lot of modeling to try to understand what the effects are. This is another study that was published in Science that tries to look at ecological processes, again, is surveying all of the contemporary work, trying to understand what species are going to be affected, how are they going to be affected, what can we do? And so this is a perspective way to look at the problem of climate change, and there's a lot of resources going into it. But there's another approach that we could take, which is a retrospective approach. And instead of looking forward, we could actually look backwards and try to understand how previous climate changes that have occurred affected species of the time, and what can we see about those in today's surviving ancestors based on their genetics. And so I'm going to tell you a story today about Antarctic notothenoid fishes. And these fishes are very interesting and weird. And the notothenoids had an adaptive radiation during the Eocene about 40 million years ago. And at that time, the Antarctic environment was not cold. It was actually cool temperate. So it was similar to what you would find around New Zealand today. And around Antarctica at the time, there were sharks. We know that from the fossil record. And so we know that there was trophic levels that could support those sharks. And however, that line of fossils that we see in the record are not the ancestors of notothenoids. So this is an example, this is a model of how the continents have shifted through plate tectonics over the last 200 million years ago. And so we can see here, I've labeled the continents to orient you. There was one supercontinent at this time, and you can see Australia up here and the tip of South America down here. And we can watch what happened over 200 million years. So you can keep your eye on Antarctica and South America here. And so over tens of millions of years, the continents split apart, and Antarctica relocated to the current position at the South Pole. And eventually it was separated from all the continents that surrounded it. And so here it is today, and here is the tip of Argentina and Chile today. And the result of this was the formation of the Antarctic circumpolar current. And so there's basically this giant current that goes all the way around the continent. And because of the density of the cold water in the Southern Ocean, it's much saltier and denser. And the warm water from the tropics moving down, it sort of forms into this ramp where it runs into this dense cold water. And then you have winds going all the way around the planet. And basically this creates a barrier. So no fishes come in and no fishes get out, at least not very easily. And so this has isolated this entire area. So as Antarctica became isolated, these ice sheets began to form, and these ice sheets basically scraped out all the benthic environments around the coastal areas and destroyed all the habitat, and pretty much all the endemic species went extinct. The exception, of course, were the notothenoids. The notothenoids essentially survived this, and because everything else was gone, they essentially evolved to fill all the remaining niches. So they had a free space to do all sorts of different things. So for example, notothenoids, most fish have swim bladders that they fill with air, lets them go up and down in the water column. Notothenoids don't have those, but they evolved the ability to colonize different parts of the water column by doing things like reducing their bone density and increasing fat deposits in their muscle. And in fact, notothenoids have been used as models of osteoporosis because they're evolutionarily parallel idea of whatever caused them to lose their bone density is the same process that humans endure as they age and we lose our bone density. All right, so this is a tree of all the Antarctic species. It's a lot to see here, but basically there's about 28 species that are outside of Antarctica, and there's about 100 or so that live around Antarctica, and they consist of the vast biomass of the ocean. So if you go out there and you fish, you're gonna find notothenoids. And we're going to be talking about this group of cold specialized fishes, and we call them cryonotothenoids. So these are all the fish that are specialized to live in this region. And how did these fish do this? So everything else went extinct. How did the notothenoids survive? Well, the basic idea is they evolved a gene de novo. So they invented a new gene, and this gene is known as an antifreeze glycoprotein. And the cool thing about this gene is it's not like antifreeze you put in your car, which is basically sugar water of sorts. This antifreeze protein will, when it comes into contact with ice crystals, it physically grabs the ice crystals and prevents them from growing. So these fish are full of antifreeze proteins that hold on to those ice crystals, and it's not actually known what happens to all those ice crystals over the life of the fish because they just sit there and they stay. But this is a really neat process. This is a gene that's not descended from another gene. It's not from a gene family. It was invented completely in the notothenoid lineage by taking what's called a trypsinogen gene and having this novel expansion of repeats in the middle of it that give it this ability. And this is not the only time antifreeze proteins have been invented. They've actually been invented a few different times in different parts of the world. Okay, so having this cold specialization has a lot of consequences. And so again, this gets back to these ideas of climate change. So if you become very specialized to the cold, what happens if it's not cold anymore? So for example, cryonotothenoids vary in their ability to mount what's called a heat shock response. So most of us, if you go into a sauna for a long period of time, your body's going to start transcribing heat shock proteins. And those heat shock proteins are going to help your other proteins fold and not melt. So if you do this to cryonototheniioids, you heat them up to what's called CT max, their maximum temperature they can take before they pass out. You can see over time, they start to transcribe a whole bunch of genes, including heat shock proteins. And that's what you see here. This is Eleganops maclavinus. This is an outgroup to the notothenioid, so this is not an Antarctic fish. And these are genes that are downregulated and upregulated after it's been exposed to these extreme heat events. And this is an ice fish. So this fish has a much smaller reaction. So you can see this over here. These bright yellows and oranges are genes that are highly changed. These deep purples are genes that are kind of sort of changed. And the ice fish, the non-notothenioid has lots of changes. The ice fish has a very small number of changes. And this is the most cold specialized notothenioid, Pagnothenia borchgrevink. And what you see here is there's no change at all. It has no heat shock response. So essentially its heat shock response has been constitutively jammed on by evolution, and it cannot mount an additional heat shock response. So if you put this fish in water that's a few degrees above what it's used to living at, it will die. So we wanted to understand these effects of when you evolve in one environment, what happens in a new environment. And so we're going to look at a particular branch of the notothenioids that are known as the icefish. And the icefish are really weird. So here are a tree of most of the ice fishes, and we're going to be focusing on this pair, Scephisophilis esox and Scephisophilis gunneri, up here at the top. And one of the most interesting things about ice fishes is they have no hemoglobin. So as you probably know, hemoglobin has iron, and it transports the oxygen around your blood so it can be delivered to your tissues. And notothenioids have hemoglobin, but not ice fishes. This particular subset of notothenioids lost their ability to transport hemoglobin. So how do they survive? So they have less than 10% of the oxygen carrying capacity compared to a normal red-blooded fish. And in fact, they're not red-blooded because they don't have hemoglobin. There's no iron in their blood. So their blood is actually clear, which you can see here. This is their heart. Their heart is, tissue colored. And this is compared to a normal fish or any of us, what we normally see. And they've had to evolve all these crazy compensatory changes. So they just have to move oxygen through their blood passively. They don't have a molecule to transport it. So they have to move way more blood. So they have huge hearts. They just are constantly pumping blood in vast volumes to keep the oxygen going. And here is the cold specialized notothenioid. That's its eye. Here's icefish. So these are the vasculature. These are the arteries and veins. You can see that the icefish just has way more arteries and veins because it has to move all this passive oxygen around. So is icefish performance limited to these narrow temperature and oxygen conditions? So we saw that borchgrevink doesn't have a heat shock response. Well, this seems way more extreme. So what would happen to this fish in a changed environment? And the really cool thing here is we know. How do we know? Well, we know because there is a sister species, but basically the icefish are hypothesized to be able to survive without hemoglobin because in cold water, there's more oxygen. More oxygen can be dissolved in cold water, and in cold places, the metabolic rate slows down. So people think, okay, you don't have to move so fast, and the water has a lot more oxygen in it, so these ice-fishes can probably survive. And when we look at this, everything has hemoglobin, and so papers would often characterize this loss of hemoglobin in this way. So as this kind of catastrophic event, like how are these fish surviving? Why would evolution do something like this? It must have just been this crazy one-time loss, and somehow the fish weren't extirpated. But it's not actually true. If you look across, now that we have more data, we've looked at a lot more ice fishes, other groups, what we can see is that hemoglobin has actually been receding in notothenioids over the evolutionary tree. So what does that mean? We tend to have multiple forms of hemoglobin in our bodies. We have different protein isoforms. So we have fetal hemoglobin, we have adult hemoglobin, and other organisms have hemoglobin for low temperature, high temperature. Basically, we produce a bunch of different kinds of hemoglobin for different environmental effects. But in the notothenioids, not just in the ice fishes, but across the clade, they've been slowly giving up their different isoforms of hemoglobin. And then if you look at this complicated image, all you have to know is that there's more bars of more types of isoforms. And when you get down to the ice fish down here at the bottom, there's no more bars. So the ice fish were the last fish to give up that hemoglobin and just assume that they don't need it. So it didn't have to necessarily be a catastrophic event. Again, there could be good evolutionary reasons to give up hemoglobin in terms of having your blood be more fluid so that it can move in cold environments in a better way. Or there is a strong relationship in the amount of iron available in the ocean, and there may have been a lack of iron and it was easier to do without it. So the question here is, has this over-specialization doomed ice fishes to the Southern Ocean and made them vulnerable to climate change? So as I started to say and stopped, how can we answer this question? Well, we have a sister species. This is the pike ice fish, Scephisophilis esox, and this fish colonized temperate waters on the tip of South America. So cryonotothenioids evolved from a temperate ancestor up here. This fish, esox, has a sister species, Gunneri, which is still found in the Southern Ocean outside of Antarctica. So we have this tree here where we have the ancestral temperate ancestor, we have this gain of antifreeze proteins, this proliferation of notothenioids, and down here on the ice fish branch, we have this loss of hemoglobin, and finally we have the escape from Antarctica. So how did this fish escape from Antarctica? The hypotheses are that during certain ice age events, when the water levels, ocean levels dipped, that changed the Antarctic's polar front enough to allow these fish to get out of Antarctica. And these two sister species are very young, only less than 2 million years ago they diverged. Okay, so esox lives in a really different environment. So here's the West Antarctic Peninsula. This is where you'll find Gunneri, and this is the Patagonian area of Argentina and Chile, and this is where you can fish esox. Okay, so our goal as a group was to characterize the genomic architecture of temperate adaptation in sea esox. So what can we find out about esox that might shed light on how it made this transition? And at the time, there were no genomic resources available for any secondarily temperate fishes, including esox. So we set out to assemble and annotate 2 genomes for esox and its sister Gunneri. And then we did a population genomics RAD-seq-based analysis to see if we could detect regions of the genome that might be under selection. Okay, so we didn't have any esox species, so the first thing we had to do was go get them. So we went down to Puerto Natales, Chile, and we fished off of the pier for a few weeks. That didn't work very well. Eventually we found some fishermen, and eventually they took us out on their boats, and that's Angel going out on the boat, and they picked up some esox in some of their nets. We were able to bring those back and extract DNA from them. And here you go. This is the genomes of esox and Gunneri, and this was done with PacBio long-read sequencing combined with high-C reads and some RNA sequencing. It all looks very simple. It took us several years, and during this time, PacBio was just being developed, so we actually tried to assemble the genome multiple times with multiple technologies, and it wasn't until PacBio that we could really put this together. But we could see these are the 24 primary chromosomes of the 2 genomes. You can see how similar they look. They're about 1 billion base pairs each, and our genome has an N50 of 44 megabase pairs, which is very good. And we have found about 28,000 genes in each of the fish. So we wanted to compare the genomic architecture of these 2 fishes directly. So this is software that we've developed in our labs called Synolog, and what we're looking at here are the 2 genomes. This is Esox on the top, Gunnerai on the bottom, and then we have each chromosome here alternating in grays. And then each line here is one orthologous gene that we're able to identify between these two genomes. And so what you can see broadly is that the genomes are very, very similar, which we would expect. There's only been 2 million years. But you can see if you stare at this for a while that there are some changes that occur in between these genomes. And here are some examples. So this is our two ice fishes, and this is the outgroup. So largely the genome is conserved with the non-Antarctic fish, but we see things like inversions. So these are pieces of the two ice fish genomes that have actually inverted several megabases in size. And you see the inversion relative to the outgroup, but not between the two ice fishes. So these pair of inversions occurred before the ice fishes diverged from their ancestors. But we do see specific inversions that occurred within Esox relative to its sister Gunnerai. So in the last 2 million years, this chunk of the genome in Esox broke off and inverted and reattached, and that's still there. And then the biggest thing we found was an entire chromosome. So this is chromosome 21. And what you can see here is the chromosome essentially split in two halves, reversed, and flipped itself. And so it's completely inverted. And so why do these things occur? Well, they probably occur all the time, but why are they maintained? Why are they selected over time? And usually we think it has to do with gene regulatory regions either being moved closer together or split apart, and those become useful in some evolutionary contexts, and that gives a selective effect to maintain these things. So we wanted to look for some markers so that we could investigate this a little more deeply. So we were able to obtain 28 total C. Esox individuals during our time in Patagonia. And from previous collecting trips, we were able to get 68 Gunnerai samples. So you can see we sampled here and here, and then Gunnerai came from here and here. And so we extracted DNA. We used a technique called rad sequencing. And in rad sequencing, again, you guys have these amazing panels of markers that have been curated for decades. We had 0 markers. We had to create markers on the fly and then use them. So what we do is use a restriction enzyme, which chops up the genome in specific places. We pull that DNA down, and then we do paired and Illumina sequencing from both directions, and we assemble a mini contig of that region. We call SNPs within that region, and then we correlate all those loci across species. And in doing that, we were able to assemble 56,000 loci from across the genome, and that gave us 288,000 SNPs. And what was cool was each locus that we were able to pluck out of the genome with our software was about 650 base pairs in length. So an Illumina read is about 150 bases. So we used some sequencing and computational tricks to put different paired end reads together so that our average locus size was over 600 bases. So that allowed us to generate some nice statistics. Okay, so we wanted to find evidence of selective sweeps between these two species. And so what does a selective sweep look like? This is, I think, very similar to the concepts that you're all looking at in crops and other places. We're looking for linkage blocks. So our genome is not transmitted in chromosomes, it's transmitted in linkage blocks. And we can imagine our two species, each with its pair of diploid chromosomes, and they have assortments of different blocks of different sizes in the population that are segregating. But when there's a selective event, when some in the environment selects on a particular set of loci, we might find a region like this where one particular block has been selected to fixation in one species, but in the other species, there's no selective effects at different environment, and so you just have the same random assortment of blocks that are segregating the population. So we're going to use extended haplotype homozygosity and DXY to search for these signals in our data. And we're going to plot it along the chromosomes, and this is what it looks like. So this is DXY. This is a nucleotide diversity measure, and this is chromosome 12 from start to end, and this is chromosome 21 from start to end, and this has just been kernel smooth. So we have those 58,000 loci across the whole genome, and so we're smoothing our measurements of DXY, and we see peaks, and sometimes those peaks by a bootstrapping procedure are found to be statistically significant. And then we look at the same markers using this cross-site extended haplotype homozygosity. And so this is looking for these extended haplotypes, so these fixed linkage blocks. And so if you see a peak going to the upwards, it's fixed in ESOX. If you see a peak going downwards, it's been fixed in Gunneri. So we're looking for peaks in the upward direction, and we can see a bunch of peaks here that are statistically significant on these two chromosomes. And what's cool, we can put all this data together with our conserved synteny. So now we can ask, what does the genomic structure look like underneath these peaks? And we can see some cool things. Here on chromosome 12, we see this inversion between our two fishes, and it's right underneath these two signals of selection. And here, where we had the entire chromosome flip and reverse itself, we again have two peaks on either side of the inversion, the split point. So that means that these peaks were here, and this peak was there. They got moved together, and something about having these groups of genes and their regulatory regions in the same place was beneficial. It was beneficial enough to keep this weird change in architecture in place. Okay, so if we zoom in here, we can look at our three pieces of data together, and we can zoom further in, and we can say, well, what genes are underneath these peaks? So in this peak, this is our extended haplotype in red. This is ESOX. And in blue, this is the Gunneri. And what you can see is there's a much bigger peak in the ESOX versus Gunneri. And we have this gene, TMRT2A, which is a methyltransferase gene. So this is a gene involved in moving methyl groups around with the mitochondria. So we're not really sure why this gene would be selected. But it's involved in RNA methylation. But in other peaks, we found genes involved in the heat shock response. That makes sense. This fish went from a very cold environment to a temperate environment. We found genes involved in mitochondrial function. So I'll come back to that, but these fish have extreme mitochondria that are really interesting. And there's genes involved in vision. Again, that makes sense. The light regime at the South Pole is basically always dark or always light. And of course, in Patagonia, you have a much more normal diurnal cycle. So it would make sense that you would see changes in vision. All right, so what do the AFGP genes look like? So again, this is what allow the notothenoids to thrive, but ESOX doesn't need AFGP. They don't need antifreeze proteins. They're no longer in below freezing water. I think I skipped over it earlier, but the Southern Ocean is minus 1.8 degrees Celsius year-round. So it's below zero year-round. But obviously the waters around Patagonia are significantly warmer. So this is the region of where that gene occurs that we assembled. And just to orient to what you're looking at, down here on the bottom, this is Eleganops maculavenus. This is our outgroup. I already showed you a couple plots with this guy. These are all cryonotothenoids. We have our ice fishes, and then these are the two new ice fish that we assembled, and this is our secondarily temperate ice fish at the top. So we can see this is a similar region, and in our outgroup, the outgroup doesn't have any antifreeze proteins. That's what we expect. They evolved after that split. But what we can see is compared to the more cold adapted ice fishes, there's many fewer antifreeze glycoproteins in these new ice fishes. And it's hard to see, but there's little size symbols on here. These are pseudogenes, so most of the gene is still there, but they have a stop codon or a part of the exon's been blown away. So you still have 3 of these intact genes in Gunneri and 1 intact gene in ESOX. But interestingly, for the first time, we found a second region containing additional antifreeze proteins. This has never been found in a notothenoids before, and it was upstream of the standard region. So some of these genes duplicated and moved, and we know that this is correct and not an assembly error. First, we have conserved synteny evidence. So again, we're looking at where the genes lie. And what we see is this is the canonical locus I just showed you. This is where all those AFGP genes are at. And we see it conserved in our 2 new ice fishes and an additional ice fish. And this is the non-canonical, the new locus, and it is also syntetically conserved. So we see the same order of the same genes in the same place in the 2 species. And that's from independent assemblies, independent annotations, and so on. And we used a software called Clumpy that was written by one of my newer students, Gio. And this is a really interesting software that's great with PacBio data. And essentially what you can do is take your assembled genome and you can take your long reads and you can give it a gene model or anything else. It could be a transposable element, anything that you want to find. And you can say, hey, find this. It will search the reference genome and then it will search all of your long reads. And so each of these is one single PacBio read, and you can see that how they were assembled together to form the chromosome. And you can see the blue squares are the exons of this gene, and they've been painted not only on the reference genome, but into the reads themselves. So you can see precisely how the structure of the genome was put together and that this locus is real. Okay, so that's really interesting. But there's more than one genome in an ice fish. There's more than one genome in pretty much every organism. Not quite all of them. But what I'm talking about is the mitochondria, of course. So the mitochondria is your power-producing unit. It has all sorts of functions for the health of your cells. And one could imagine without hemoglobin, and in the cold that you could need some heavily modified mitochondria. And that's indeed the case in ice fishes. So ice fish oxidative muscles, like their heart, they have to have really high densities of mitochondria. They don't have to, they do. We've observed that they have them. And that makes total sense because their heart has to work much, much harder, pumping blood all the time. It needs a lot more ATP to power the muscles to do that. In fact, they have so many mitochondria, it's similar to the number that hummingbirds have in their wing muscles. And it's greater than the swimming muscles of tuna. So if you know anything about tuna and their ability to power across the oceans. So we have a lot of mitochondria in these fishes. And this is just showing you, this is an electron picture from an electron microscope. And so this is a red-blooded notothenoid, and this is one, I've highlighted one mitochondria here in the image. And in Esox and Gunneri, in the ice fishes, you have these gargantuan mitochondria. So they're just vast. And even though they're much larger, their intermitochondrial membrane, which is where the cristae sit, which is where your complex 1 through 5 is, which is where you're producing your ATP, they're roughly the same. And there is a hypothesis that they have these enlarged mitochondria in order to compensate for their lack of hemoglobin and myoglobin. So I didn't mention that, but most of them also lack myoglobin. And one hypothesis is that they're storing, essentially accumulating oxygen in these big regions to just keep it around for whenever they need to use it. So we, talking with people and understanding the physiology of these fish, we decided to look at the assembly of the mitochondria to see what might be happening. But we didn't actually target the mitochondria. Actually, when you sequence a genome, the goal is typically to exclude the mitochondria. There's lots and lots of mitochondria, so if you extract DNA from a sample, you don't want to sequence 99% mitochondria and waste a bunch of money. So we just looked at our sequencing libraries that we built from nuclear DNA. So we said, maybe we got some data by accident that snuck through, so let's just search and see what we found. And we did. We actually got quite a bit of data from both of our ice fishes that were just hidden in the nuclear data. And so we searched it for typical mitochondrial genes, we extracted it, and then we were able to assemble it and annotate these mitochondrial genomes. And this work was led by Bushra Minhas, another one of my students, and Emily Beck, our collaborator from Oregon, who just started a lab at the University of Kansas. And one of the cool things about PacBio is that it can produce in a single read the entire genome of the mitochondria. So our PacBio reads could be anywhere from 15,000 bases to over 75,000 bases. So we could get multiple reads from the same mitochondrial genome and compare them. And there was no assembly per se in this process. But what we have to remember is that there's not a single mitochondrial genome. I mean, you have orders of magnitude more mitochondria in the tissues than you have nuclear DNA. So when you're getting multiple copies of that genome, are they all the same? And we took a look at it. And so here is, this is zebrafish, Danio rurius. This is the standard model in the fish world. And this is what its mitochondria looks like. It's actually very similar to the human mitochondria. And you have a bunch of protein coding genes, some tRNAs, this control region, which is where a lot of regulation occurs, and a few other bits and pieces. But the whole thing is, I don't know, about 18 kilobases in length. So it's very small. And we want to focus on this gene ND6. ND6 is a really weird gene. And like if you start searching the literature for it, you find all kinds of weird things. So for example, it's been characterized as a disease-causing gene in the human brain. So it's been associated with neural diseases. It's a source of mutation from arsenic exposure. So they've looked at humans that have been accidentally exposed to the high amounts of arsenic, and they find all these mutations in the ND6 gene. It's a source of evolutionary adaptation to high altitude in horses. So they got looked at there. It's also been cited as a source of innovation in human tumor cells. So you look at tumors, you find weird mitochondrial stuff, and it often involves the ND6 gene. And so we looked at our assembled mitochondrial genomes. And we found essentially that ND6 had been moved closer to the control region. So it had been moved closer to the area where transcription starts in the mitochondria. And not only that, we found these crazy results that you see here. So this is Gunneri on the left and Esox on the right. So Esox is the secondarily tempered fish. And in Gunneri, we see this segmental duplication. So here's ND6, here's ND6 again, here's ND6 again, here's ND6 again. And the control region has been duplicated 4 separate times. And in Esox, it's even weirder. We have the original unmodified version. And here we have a version with ND6 3 times with 3 control regions. Here's the ND6 4 times with 4 control regions. And this occurred in the same fish. So this fish has different mitochondrial genomes segregating in its body. So how can that be? So if you know about the mitochondria, it's passed down in the maternal line. It's a maternal marker. It gets put into the egg. Sometimes a few mitochondria can sneak in from a sperm, but before the mitochondria can get into the egg, there's this wild selection process where the body gets rid of, I don't know, 90% of the mitochondria, and only super special mitochondria get into the egg. So where do these mutations occur? Are they segregating the population? Are they evolving de novo within the life of these fish? And we don't know because we weren't looking to sequence the mitochondria. This was just a byproduct of the reference genomes. But we kept looking. So once we found that, we just went out to the data banks and started downloading any PacBio data set from any notothenoid, and we found more weird stuff. So here are 2 other ice fishes that again have multiple segmental duplications or multiple, this is called heteroplasmy, when you have more than one genome segregating. And we've even found in another cold specialized notothenoid, the gene had moved to the other strand. Anyways, just all sorts of weird stuff is going on. So in the ice fish, what is the exact function? So we don't know, but we speculated and looked into it as best we could. And so here is the 5 major complexes of the electron transport chain. And that's part of the oxidative phosphorylation system. So this is where you're getting your ATP from. And most of these complexes have a mixture of genes that are encoded mostly by the nuclear genome and some by the local mitochondrial genome. And this is where reactive oxygen species is produced. So when you have to work really, really hard in your mitochondria, you're going to be shooting out electrons that can go and damage other things. And a lot of the physiology around mitochondria is how do we control reactive oxygen species. Okay, so complex 1 is here, and complex 1 has the most mitochondrially encoded proteins of the 5 complexes. So if you're going to make it directly in the mitochondria, it's going to be involved in complex 1. And here's a schematic of ND6 embedded in complex 1. And this is a human example. So there's 5 different domains that come out of ND6. In zebrafish, the model, there's the same 5. Here's an icefish. Here's 2 icefishes with the same 5. They all look the same. Now, when we looked in ESOX, we have 2 different versions of complex 1. So here we have the normal one. So here we have a intact version, and here we have a truncated version. And in Gunnerai, we have but it's the opposite truncation. Okay. So of the 5 domains, in ESOX, we have 1/2 of them, and in Gunnerai, we have the other half of them. And so why is that important? Well, if we look at complex 1 and how it embeds into the cristae, this is all of complex 1 here. You can see there's lots of proteins here. And what you have is a peripheral arm and a membrane arm, and this is a hinge point between these two, and these are on different sides of the inner membrane matrix. And ND6 is right at the hinge point. So ND6 controls how this hinges it embeds across the inner matrix. And so when you have these 2 different truncated versions, we think that that's affecting the hinge point in complex 1. Okay, so this is as far as we could take it, but it's opened up all kinds of questions. And basically where we're trying to take this research now is to do a direct examination of mitochondria across notothenoids. So for example, can you have tissue-specific mitochondria? Can you have that the heart has one type of mitochondria that's specialized for one type of energy production, but the brain, where you need to resist reactive oxygen species, might have something different? Is it segregating in the population? If we can sequence eggs directly from these fish, which we're trying to do, what is the distribution of mitochondria that we see there? So there's a lot of questions about where and how these changes are occurring, but this is as far as we could take it, given the fact that icefish are really hard to get. All right, so just to sum up, the large scale, so we looked at these 2 icefishes, one that's still cold specialized, one that's escaped and lives in Patagonia. Their genomes are largely maintained, but we do see big structural changes in specific places, and we see using markers that there are genes there that appear to be under selective constraints, and those genes seem to be associated with the structural variants. The antifreeze proteins still exist. There's fewer of them. Many of them have become pseudogenized. That's what you would think would happen in a warmer environment. But there's an additional locus of more AFGPs. And why are those there? We're not entirely sure. Selection accelerated lineage sorting between these Antarctic and temperate adapted alleles. And we see this novel mitochondrial genomic architecture, so extensive heteroplasmy. And we know from other work that there are several nuclear encoding, including our own work, that there are several nuclear encoding mitochondrial genes under selection. So I showed you one of our genes that was mitochondrial related, and then I showed you mitochondria directly that were all different. So maybe all those things are working in concert to adjust their physiology to this different environment with no hemoglobin. So that's my story of adaptation following adaptation from a highly specialized ancestor in the last 1.6 million years. So thank you for your time.
We now have some time for questions. So
Very nice talk. I really, going back to basic biochemistry, did you try to isolate the matrix and try to measure the ATP formation?
No, but that you're basically, we put in a grant proposal last month that asks to do that. So I'm a humble computer scientist, so that is way beyond my capability. But my colleague at Emily Beck, she will hopefully be able to do that soon.
Yeah, because that might be very interesting to see if the mitochondria can produce ATP as fast as any other mitochondria.
Yes.
And the same on the other side, glycolysis would be something to measure.
Yeah.
You see, and to see if you are, basically is doing fluxomics to see how the mitochondria is being fed by metabolites and see how the ratios are going, because these fish they don't need to have much to spend much energy, right? They are in a very cold environment, plenty of oxygen, and the other thing would be to measure the oxygen ratios, the inside.
Yeah, I agree with you 100%. So I think we just have to get much deeper into the physiology and the chemistry.
Yeah, the physiology is something remarkable.
Yeah.
I would never expect 2 or 3 different genomes inside the mitochondria. It's fantastic.
Yeah, when your student comes to you with 2 or 3 genomes, you say,
you did something wrong.
Did you check that twice?
You contaminated the sample.
Yes.
Very nice. Thank you very much.
Thank you.
Is it possible to compute the frequencies of the genomes? So how many times do you find any, like the 2 copies versus the 3 copies?
Well, yes. So, I don't know how easy it would be to come up with a global number. You'd have to normalize it by like number of cells. I think the easiest way would be in an egg, because if you could single cell sequence an egg, then you have a much more fixed quantity. But I think I really want to do tissue specific sequencing and
otherwise there. If you don't, it can be just a somatic mutation, right? Like you find ones.
Yeah, or one hypothesis is that the maternal line's clean, that there's only one genome and there's nothing segregating, but these are occurring de novo. Like once you get to developmental stage X, you start to get mitochondria drifting in different directions because it's physiologically good, but we don't know. So yeah. Okay.
Thank you, Professor Catchen. On the behalf of the organizing committee, we appreciated your presentation and for sharing your expertise with us. And to express our gratitude, we have a small gift for you too. So